Hello, my name is Sidrah Ashrafi, and the focus of my research is to characterize the role of the transcription factor interferon regulatory factor 5, or IRF5, in iron homeostasis. IRF5 is a transcription factor that is known to play a role in the immune response, which is how our body protects us from bacteria and pathogens. IRF5 specifically is associated with the toll-like receptors, which are a family of receptors known to help mediate the inflammatory response. A pathogen binds to the toll-like receptor and initiates a signal transduction cascade that prompts IRF5 to translocate to the nucleus, where it promotes the production of pro-inflammatory genes and cytokines. Multiple studies have placed IRF5 as a central regulator of the inflammatory response, and the gene contributes to the pathogenesis of autoimmune diseases, including rheumatoid arthritis and inflammatory bowel disease. Additionally, IRF5 has been seen to have multiple or pleiotropic effects, and dysregulation of IRF5 contributes to the development of asthma, neuropathic pain, and cancer. Recent preliminary data from my lab, the Blanc Lab at the Feinstein Institutes, has identified a potentially novel role that IRF5 may hold implications in. It was seen in 3-month-old mice with a complete knockout of the IRF5 gene that these mice had reduced red blood cell levels and reduced hemoglobin levels, contributing to an anemic phenotype to develop within these mice. This suggests that IRF5 may hold implications in red blood cell productions, and therefore my project focuses on the region in which red blood cells are produced. Red blood cells are produced within the bone marrow with the help of specialized cells, the most important one being the erythromyeloblastic island. These islands consist of a central macrophage surrounded by developing red blood cells, and the job of the central macrophage is to provide these blood cells with nutrients as well as to extrude the nuclei of mature red blood cells, but most importantly maintain the balanced trafficking of iron needed for red blood cell production. Macrophages specifically are known to play a key role in iron homeostasis as they express various iron regulatory proteins that maintain the balanced trafficking and storage of iron in and out of the cell through an accessible pool of iron ions known as the labile iron pool. It was seen that a loss of IRF5 contributed to an anemic phenotype, and therefore my project investigates how a loss of IRF5 may impact iron homeostasis, and I hypothesized based on preliminary data that a loss of the gene would result in dysregulated iron homeostasis. I first wanted to see how IRF5 affects the labile iron pool of macrophages, and in order to do this, I used bone marrow-derived macrophages from both wild-type mice and IRF5 knockout mice provided to me by my mentor. I then cultured these cells for 7 days and measured their intracellular iron levels through a protocol known as calcium AM staining. Calcium AM is a cell-permeable molecule that upon entering the cytosol interacts with cytosolic proteins known as esterases. Through hydrolysis, esterase cleaves the AM portions of the calcium, and this results in fluorescence, and for this reason calcium is often used as a live dead stain. However, the presence of iron within the cytosol is known to inhibit this interaction. But by introducing an iron chelator, the activity of iron is restricted. Therefore, after treating my cells with calcium, I split them into 2 groups, a control group and a chelated group, and measured the difference between their fluorescency. This fluorescency represents the amount of labile iron present within the cell. It was seen that with a simple knockout of the IRF5 gene that the wild-type macrophages had relatively normal iron levels, but the knockout macrophages had reduced iron levels. I wanted to investigate this further and see if this reduced iron pool could be remediated with an iron supplement, and in order to do this, I treated my cells with ferric ammonium citrate, which is often used in studies as an iron supplement. I treated my cells with specifically 10 micromolars and saw that the wild-type macrophages were able to uptake the iron and their iron levels increased, but the knockout macrophages still had reduced iron levels. I then wanted to investigate this further and see if perhaps stimulation is required in order for the macrophages to uptake iron. In order to do this, I treated my cells with lipopolysaccharide, or LPS. LPS is known to interact with the toll-like receptors, and these receptors specifically are what are known as pattern recognition receptors. These receptors recognize preserved regions of pathogens known as the pathogen-associated molecular pattern, or PAMP. LPS is a specific PAMP of gram-negative bacteria and known to interact with the toll-like receptors. So LPS binds to the receptor and initiates a signal transduction cascade that prompts IRF5 to translocate to the nucleus. So perhaps by stimulating the gene, it may help the macrophages to uptake the iron. I first treated my cells with LPS alone to see if it would have any effect on the labile iron, and it closely resembled what it looked like with a simple knockout of the gene, and that the wild-type macrophages had relatively normal iron levels and the knockout macrophages had reduced iron levels. I then treated myself with both the LPS and the iron supplement and saw that the wild-type macrophages were able to uptake the iron, but the knockout macrophages still had reduced iron levels. This suggests that the mechanism to help regulate iron levels is affected by a loss of IRF5, and in order to investigate this, I performed RNA sequencing analysis. RNA sequencing is a method often used to measure the differential expression of genes. I specifically used a publicly available RNA dataset from this study that looked at IRF5 in relation to obesity. However, this study did not characterize any iron regulatory proteins. In order to analyze this dataset, I used integrated differential expression pathway analysis, or IDEP. And through IDEP, I did specifically what is a PCA, or a principal component analysis. Through this component analysis, I was able to see that there was distinct clustering between the knockout and the wild type groups. This means that there was distinct differentiation between the control group and the experimental group, and that fair comparisons could be made between the two. I went through the dataset to see if any iron regulatory proteins expression was affected by a loss of IRF5, as there are various iron regulatory genes that have been characterized in macrophages to play a role in iron. I identified 7 genes whose expression was affected by a loss of IRF5, and so the altered expression of some of these genes may be a cause of the low labile iron pool, and some of these genes may be an effect of the low labile iron pool. It was seen with the loss of IR5 that the expression of transferrin was downregulated. Transferrin plays a role in incorporating iron into the cytosol, and this may help explain the low labile iron pool that was seen, as less iron is being incorporated into the cell with the loss of IR5. Another gene whose expression was affected includes midopterin-1, which helps to incorporate iron into the mitochondria. It was seen that with the loss of IR5 that this gene was upregulated, which suggests more iron is being incorporated into the mitochondria and less iron is present within the cytosol. Other genes whose expression was affected includes SLC11 and FLVCR, which are both expressed on the membrane of macrophages and help to incorporate iron into the mitochondria. Other genes whose expression was affected includes ferritin and NCOA4, which are both involved in iron storage. Both of these genes were downregulated with the loss of IR5, and this could be because there's already low labile iron present within the cell. Another gene whose expression was affected includes SLC40, which is the major iron exporter of the cell. This may be an attempt by the macrophage to preserve the small amount of labile iron present within the cell. Future studies would be to determine the total iron content of the cells, as well as to measure the intracellular and extracellular iron levels, and to verify the expression of the genes identified from the RNA sequencing using qRT-PCR. In conclusion, the results from the study identified for the first time a role for IRF5 in red blood cell production and iron homeostasis. The results from this study provide important insights into the molecular mechanisms that may define the interplay between the central macrophage and developing red blood cells. Understanding the pathways that regulate red blood cell production is essential for the development of new therapies to help cure anemia, a disease that currently affects 1 billion people worldwide. These are my selected references. Thank you for listening to my presentation.